Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were discussing about cellular signaling, in which we have covered multiple signaling pathways that are seen in the biological systems, which can be mediated by the cell surface receptors, the different types of cell surface receptors, such as the ones which are being coupled with G proteins, the G protein coupled receptors, the enzyme coupled receptors, in which the receptor tyrosine kinases is the biggest group. So we discussed about receptor tyrosine kinase mediated, or if the receptor tyrosine kinase gets activated, what types of signaling that is the cell is going to follow. We discussed about that. Then we discussed about some enzyme coupled receptors such as cytokine receptors, which are being coupled with Janus kinases to do the downstream, to activate downstream STAT proteins. So we discussed about such pathways. Then we discussed about paracrine signaling pathway, where the notch and delta receptors, they are interacting with each other physically to mediate the notch responsive gene transcription. So we discussed about that. Then we discussed about Wnt signaling pathway, the TGF beta signaling pathway. And today we are going to discuss about a very important and unique pathway known as Hedgehog signaling pathway. So this Hedgehog signaling pathway is named over the protein Hedgehog because this protein Hedgehog is going to it is going to behave as an as an extracellular signal protein. So this Hedgehog protein, it is going to activate some receptors which are going to downstream activate genes which are known as Hedgehog responsive genes, and that is why this signaling pathway is referred as Hedgehog signaling pathway. Okay, so this Hedgehog protein was first identified in Drosophila. So in Drosophila, when a mutation of certain gene was seen, it resulted, the mutation in the gene which encoded for this Hedgehog protein, it resulted into the formation of spiny processes over the larva of Drosophila. So let's assume that this is the larva of Drosophila. So over the cuticle of Drosophila, it developed a spiky processes which are known as denticles. Okay, which gave the Drosophila, adult Drosophila, the fruit fly, the look of hedgehog. So it resembled a lot with hedgehog, and that's why this protein was named as hedgehog, because the mutation in hedgehog gene, which encodes this hedgehog protein, resulted into development of tentacles over the cuticle, over the surface of the larva of the fruit fly, and that is why this protein was named as hedgehog protein. So this gene, so that there are different types of hedgehog proteins that can be seen in vertebrates. So that the types of hedgehog proteins that can be seen in vertebrates are sonic hedgehog. So hedgehog can be abbreviated as capital H small h. So sonic hedgehog. The other type of hedgehog protein that can be seen is desert hedgehog, and third type is Indian hedgehog. So those different type of hedgehog proteins can be seen in the vertebrates. So this sonic hedgehog gene is going to encode for sonic hedgehog protein. So these are the three different types of hedgehog proteins that can be seen in vertebrates. So the hedgehog signaling pathway in the Drosophila and in mammals, it is quite similar but yet different, because the transcription regulators that are going to get activated by the hedgehog signaling, that are different. Okay, so in case of Drosophila and in the case of mammals, so they are named differently in the case of mammals and Drosophila. That is why I'm going to explain this pathway first in mammals and then in Drosophila. So this pathway, how it occurs, how it is done in the mammalian cell, and how it is done in the Drosophila cell. We will discuss that separately so that the it will be convenient to you all. So the thing I wanted to explain, that if you don't know about Wnt signaling pathway, please go and watch it, because this hedgehog signaling pathway resembles a lot with the Wnt signaling pathway. The manners in which they both of those pathway resembles are, first, both of those pathway, they are going to result into cell proliferation. So if the excessive hedgehog signaling, just like the excessive wind signaling, both of them, they are going to result into the progression of cancer, because they are going to result into cell proliferation. So if the things goes out of the hand, hedgehog signaling increases, the excessive hedgehog signaling will result into excessive cell proliferation, resulting into tumor progression, right? So this is the similarity between wind signaling and hedgehog signaling. The other similarity between both of those signaling pathways is that the presence of the extracellular molecule, extracellular signal hedgehog or wind, that is going to result into the switching on of the transcription. So this is the switch which is going to result into the switching of the transcription repression to the transcription activation. Okay, so in the Wnt signaling, the Wnt responsive genes, they were being silent, they were being repressed with the help of repressor. In the absence of Wnt and the presence of Wnt, it switched the transcription repression to the transcription activation of the Wnt responsive gene. In the same way, in the hedgehog signaling, the hedgehog responsive genes, means the genes which should be active in the response of hedgehog, which were being repressed in the absence of hedgehog. So this is a switch from transcription repression to transcription activation, just like in the Wnt signaling. So the signaling is also resembles in the manner that there exists a co-receptors known as IHOG in the Drosophila. So remember, in the Wnt signaling we discussed that LRP is the main receptor which will be activated, and then there's a receptor known as frizzled, which is a 7-pass transmembrane receptor, which resembles a lot like to the GPCR. So both of the receptors, when they get activated, then the Wnt signaling, the downstream intracellular signaling cascade, will be seen in the due to the Wnt. Okay. In the same way, in the hedgehog signaling pathway, there exists a protein known as IHOG, which is also a co-receptor for hedgehog. So the hedgehog protein, this is being received by a protein known as patched. Okay, so the patched is the receptor protein for the hedgehog signal, and the co-receptor is IHOG. That is why this resembles a lot to the Wnt signaling. So the as I'm going to explain this hedgehog signaling pathway, you're going to get the Wnt signaling pathway. So you will be able to relate both of the signaling pathways. Okay. So let's discuss about hedgehog signaling pathway, how it is done in the mammals. So we will discuss first the hedgehog signaling pathway in mammals. So this hedgehog signaling pathways, depending upon which cell is following this hedgehog signaling, depending upon the need of the cell, so depending upon cell type, as well as depending upon the developmental stages of the individual, whether it is in the embryo state, whether it is in the early developmental stages or late developmental stages, or whether it is in the adult stage and the hedgehog signaling is getting activated. Depending upon that, the hedgehog signaling pathways are diverse. So they can mediate autophagy, they can mediate apoptosis, so they can mediate a lot of factors inside the cell depending upon cell type and developmental stages. But there exists one canonical pathway that we are going to discuss today. So the canonical pathway of hedgehog signaling is going to result into the transcription activation of the hedgehog responsive gene. So keep this in mind, that the canonical pathway of hedgehog signaling will activate the hedgehog responsive genes by the activation of the patched receptor, or by the not the activation, by binding to the patched receptor. So there are multiple proteins that are going to involved in this whole signaling pathway. That is why, for avoiding the confusion, I'm going to list down all of the different proteins, all of the different factors or proteins that are present, that are going to work in the signaling pathway. The very first is hedgehog protein. Okay, as discussed earlier, hedgehog protein is the extracellular signal here. So this is extracellular signal molecule, means this is going to be produced by some certain other cell, and this is going to be received by the cell. So this is going to be produced by the source cell. Source cell is the one which is going to produce the extracellular signal, and this will be received by the sink, means the target cell. So target cell will be receiving this hedgehog as the signal. So the target cell target cell for to receive the signal, so target cell will need to have a receptor protein. So the receptor protein that target cell will have is the patched. So the patched protein, so the patched 1 and 2 in the mammals is going to work as the receptor protein. Okay, so I'm discussing the proteins that are found in mammals only. Okay, so I'm not discussing about Drosophila here. So these are the proteins which are found in the mammals. So the hedgehog protein is the extracellular signaling protein or signaling molecule, and the patch protein is the one which is going to receive this hedgehog. So that is present over the plasma membrane of the target cell. Okay. The third protein that you need to know is smoothened protein, which can be abbreviated as SMO. So this smoothened protein is also a receptor protein, okay, and it is a transmembrane protein which resembles a lot with GPCR, or you could say that it resembles to frizzled. So it is a 7-pass transmembrane protein, okay, and that is why it resembles to the GPCR. But why I'm saying that it resembles to frizzled? Because this smoothened protein, this has extracellular domain which can bind to the lipid molecules. You remember, frizzled receptor is able to bind to the lipid molecule, and that is why Wnt, which is a fatty acid chain, which has a fatty acid chain covalently attached to it. So this is the Wnt protein, and to this Wnt protein, the fatty acid chain is covalently attached. So this fatty acid chain is being received by frizzled receptors, which is a 7-pass transmembrane protein, and this particular Wnt protein is being received by LRP. Right? I hope you remember that. So in the same way, the hedgehog protein is also a protein which gets modified, and to this protein a covalently, so a fatty acid chain will be bind to this will bound this to this protein, will get attached to this protein covalently. So this is the hedgehog protein, to which a fatty acid chain can be covalently attached, and alongside, so just like fatty acid chain, cholesterol is also covalently attached to this hedgehog protein. So this is also a similarity between Wnt protein and hedgehog protein, and that is why signaling pathway resembles in a way. So the Wnt protein, it is covalently attached to fatty acid chain. Hedgehog protein is covalently attached to fatty acid chain as well as cholesterol. Okay, so this cholesterol, the way frizzled receptor is able to bind to the cholesterol, so frizzled receptor is able to bind to the lipids. In the same way, the smoothened protein resembles to frizzled protein because it has the extracellular domain which has affinity for lipids and specifically cholesterol. Okay, this, please remember this point, that the smoothened protein has affinity, it has extracellular domain which has affinity for cholesterol, because this is going to be very important to know about the patch and smoothened interaction, how they have, what kind of dynamics the patched and smoothened proteins they have with each other. So patched, actually it inhibits smoothen in the absence of hedgehog. But right now I'm not discussing that. I'm just discussing all of the different protein molecules that are involved in the signaling pathway of the hedgehog extracellular signal. So there's hedgehog, which is extracellular signal, patched, the receptor protein for hedgehog, and smoothened, which is a different protein which is present, which could in the mammals, it is present over the plasma membrane. So it is a 7-pass transmembrane protein which has an extracellular domain which has affinity for cholesterol or for lipids. Okay. The fourth protein, which is very important, is gli. So this gli proteins, so this is a whole family of proteins. So this gli family proteins, such as the gli1, gli2, GLI3. So those are different proteins that belong to GLI family. So those GLI family proteins, these are the proteins which are the transcription regulators. So these are the transcription regulators. Okay, then there exists other protein, the kinases obviously, which are going to get involved in the signaling pathway. They are pKa, CK1, this casein kinase 1 that we discussed in Wnt signaling pathway, and the GSK3. Okay, so this different these different kinases, they will also be involved in this Hedgehog signaling pathway, and there exist co-repressors such as co-receptors such as CDO. Okay, co-receptors. So there is exist other uh, proteins such as CDO, BOC, GAS1. So these are the co-receptors. Remember, I told you about IHOG. So IHOG is the co-receptor molecule for hedgehog in the Drosophila. But in the case of mammals, the other proteins such as CDO, so they are nomenclated differently because they are different from the Drosophila. So they are not conserved. So here you can say that the co-receptor molecules for hedgehog protein, they are not conserved in the mammals and in the Drosophila. So these are the different proteins, CDO, BOC. These are the co-receptor proteins that are present in the mammals. In the case of Drosophila, it is IHOG. So I will first so it will be better to list down all of the molecules because it will avoid the confusion. So alongside those molecules which have their distinct job, I will also list down the molecules which have same job in the Drosophila. So we'll form a table. So all of those molecules, all of those proteins, they are found in mammals, but they have some job, right? And that job is done by either it could be done by the same protein in the Drosophila or it could be done by different proteins. So I will be listing down all of those proteins that are present in both mammals as well as Drosophila. So these are the requirements of the different types of proteins in the mammals for the hedgehog signaling. So in the case of the Drosophila, so this was in mammals. So in the case of Drosophila, the proteins that are required that are going to do the job, they could be same or they could be different. So for example, the extracellular signal molecule is same in both mammals and Drosophila. It is hedgehog. And that is why it is referred as hedgehog signaling. Okay, so the signal molecule is same. So hedgehog is going to start the signaling. So it is the extracellular signal molecule. But the receptor which is going to receive the hedgehog, again it is same. But the proteins that will be there, they have a bit different conformation. For example, patched 1 and 2, they are present in the mammals, while there exists only one kind of patched receptor in the Drosophila. But whatever, the patched receptors are the receptor proteins for the hedgehog. So this is the receptor protein for hedgehog molecule. Okay, the smoothened also exist in the Drosophila. So all those three proteins are same, but instead of the transcription regulator belonging to GLI family, in the case of Drosophila, the transcription regulator which is going to regulate the transcription of the hedgehog responsive gene that are specific for the Drosophila, that are species specific. So those transcription regulators, they are CIs, the cubitus interruptus. Okay, so in the case of mammals, it is GLI family members, while in the case of Drosophila is cubitus interruptus, CI. Have you heard about CI yet? If you don't remember, go back to the epigenetic series. You can clearly remember CI. So the CI, this is a transcription regulator which is going to it is going to regulate the transcription of hedgehog responsive gene in the Drosophila. In the mammals, this job is done by GLI proteins, GLI1, GLI2, GLI3. So So here you can see the difference between Drosophila and mammalian hedgehog signaling. The kinases, they are same. They are going to work in the signaling pathway. They are CK1, so casein kinase 1, PKA, protein kinase A, and GSK3, glycogen synthase kinase 3. Okay, so those are the three different kinases that will be working in this hedgehog signaling pathway, and the co-receptor in the Drosophila is IHOG, as discussed before. The interference hedgehog is known as IHOG. So IHOG works as a co-receptor in the case of Drosophila. So let's now discuss about the signaling pathway, okay, the canonical signaling pathway that is followed by hedgehog protein, hedgehog extracellular signal by the cell in the mammals. Okay, so we are going to discuss about the hedgehog signaling pathway in the case of mammalian cell. So please follow this part properly, carefully, because this is going to be important in the case when I will be discussing the Drosophila part as well. So in the case of mammals, there exists primary cilium. Okay. So if you don't know about primary cilium, let's first discuss about it. So primary cilium is actually a protrusion. Okay, it is a protrusion of plasma membrane in the mammalian cell. Okay, and it is very important, it is very crucial for hedgehog signaling. It is that crucial if the, let's say, like due to certain mutation, the cell, the mammalian cell, which is going to react to the hedgehog signal, it doesn't form the primary cilium. Let's assume that there is some mutation, and due to that it is not able to form the primary cilium. If the primary cilium is not there, the hedgehog signaling won't work. So the existence of primary cilium in the case of mammalian cell, it is very important. Okay. So what is primary cilium? Primary cilium is the protrusion of plasma membrane in the mammalian cell. Let's assume that this is the mammalian cell. Okay. So this is normal mammalian cell. So in the mammalian cell, let's assume that this particular part of plasma membrane protrudes out. So let's assume that this happens. So this protrusion of plasma membrane is known as primary cilium. Okay. So that is primary cilium. So this primary cilium is formed due to the microtubule arrangement as well. So this is the plasma membrane. This protrudes out. Okay. It protrudes out. Okay, and that forms primary cilium. So in the central axis of primary cilium exists the microtubules. Okay, so it has microtubular arrangement. So you remember the when we discussed about microtubules arrangement in cilia and flagella, they were the motile microtubule arrangement. But in the case of primary cilium, this is non-motile. Okay, please listen to this point carefully, that the microtubular arrangement in the center, at the central axis of primary cilium, it is non-motile. Okay, so in the case of primary cilium, the microtubular arrangement is non-motile. So why does this microtubular arrangement exist? Because I hope you remember that microtubular arrangement is not just necessary for the movement of cell, but it is also necessary for the movement of signaling molecules. So for the movement of signaling molecules that are present inside the cilium from here to here, so from the tip of the primary cilium towards the inner part, towards the cytosolic part, that is done with the help of microtubules. So microtubules not just help in the movement of cell, but it also helps in the movement of organelles. Remember that I discussed that in the microtubules lecture in the cytoskeleton part of the cell biology. So microtubular arrangement here is important for the movement of signaling molecules that are present intracellularly. So the signaling molecules that will be assembled on the tip of the primary cilium due to the microtubule, they will be moved over the inner side, over the inner side of the cell. Okay, so that is why primary cilium is crucial, because all of the sensitivity and efficiency of any type of signaling could be increased if the signal molecules, they are being assembled in the small compartment, in the small part. You can imagine it something like this. Let's say that there are 5 or 6 balls. They are arranged in a row, and you have a wooden plank. So you have a wooden plank, something like this, and you hit the all of the balls with the help of this wooden plank, and they will be now scattered here and there. So the scattering will result into signaling which is less efficient. But let's assume that you put all of the balls inside a bag, and now you empty the bag. So you kind of shoot the bag, you kind of disrupt, you kind of blast the bag. So now this is going to result into the concentrated signaling. So this happens same inside the cell. If let's say if we have to increase the sensitivity of certain signaling pathway, or we have to increase the efficiency of certain signaling pathway, what the cells do, they just accumulated all of the signal molecules, either the signal molecules or all of the signaling components that are required, the intracellular signaling molecules. They arrange them inside the small compartment. You remember in the case of neurobiology, when we were discussing about the signal transmission from the presynaptic terminal to postsynaptic terminal, we discussed that the synaptic, the neurotransmitters, they are being concentrated in the synaptic vesicles, so that when the vesicles they fuse with the postsynaptic terminal, with the membrane of postsynaptic terminal nerve cell, the neurotransmitter that was concentrated, that was in higher concentration, they can directly fuse, they can directly enter in the cell. So this compartmentalization helps a lot for increasing the efficiency or for increasing the sensitivity of any signaling pathway. So in this case, primary cilium also does the same job. What does it do? It assembles all of the components that are required for Hedgehog signaling in this particular small compartment. And that is why the Hedgehog, the sensitivity of Hedgehog signaling increases in the case of mammalian cells. So mind this point, that primary cilium is only present in the mammalian cells for the Hedgehog signaling, not in the bacteria, not in the Drosophila. Okay, so the primary cilium has microtubular arrangement in the center, and it is going to result into the increase in the sensitivity and efficiency of Hedgehog signaling. So in this primary cilium, the membrane of, so this particular part is the membrane of primary cilium, and this is the cellular membrane, the plasma membrane, right? So over the cellular membrane part of the primary cilium exists the receptor, patched receptor. Okay, so here exists the patched receptor. So patch receptor is a large transmembrane receptor, and it has interesting qualities. Okay, it resembles a lot with the bacterial efflux pumps. And what does bacterial efflux pump do? They just remove the molecules from inside from the bacterial cell, and they just throw them out in the extracellular environment. In the same way, patched receptor, it resembles the R&D receptors over the bacterial membrane. Well, that's not the point of discussion here. The patched the patched receptor, it is the large transmembrane membrane, and it is actually 12 pass transmembrane protein. Okay, so this is the patched receptor, is a large transmembrane protein. So this large transmembrane protein has a distinct quality. What is it? It can transport cholesterol molecules. Okay, so it can transport cholesterol molecules from the cell membrane. So cholesterol, you remember when we were discussing about plasma membrane in cell biology, we discussed that the plasma membrane also has cholesterol embedded in the lipid in its lipid bilayer, right? So what does patched receptor do? This patched receptor, when it is activated, when it is in its activated form, it opens the transport, it allows the transport of cholesterol molecule from the cell membrane either to the extracellular environment, or it can it can throw those cholesterol molecule in the endosomes. Okay, so in either of the cases, what is patch receptor is doing? Patch receptor is transporting the cholesterol molecule, which is going to decrease the concentration of cholesterol molecule in the plasma membrane. Okay, or over this ciliary part, over this primary cilium part. When does this happen? When the patch receptor is active. Okay, so let me rephrase this whole thing in single sentence, that when this patch receptor, which is a large transmembrane protein, which is residing over the plasma membrane of the primary cilium, the cell part of the primary cilium, So over this membrane, this patch receptor resides, and when it is activated, okay, when is it activated, it is going to transport the cholesterol molecules from the cell membrane to either into the extracellular matrix or into the endosome, which is going to result into decrease in the cholesterol concentration of in the plasma membrane. Okay, so patch receptor, it is active, then it is going to decrease the cholesterol concentration in the plasma membrane, right? I hope you get this point. So there exists smooth and receptor over the cell membrane, which is not a part of primary cilium. So let me just draw it in the different colors. So this is smooth and receptor. Okay, and as I told you, smooth and receptor is it resembles with frizzled or GPCR, means it is a 7-pass transmembrane protein, and it has the extracellular domain. Let me just draw the extracellular domain like this. The extracellular domain is able to bind to the cholesterol, and the cholesterol binding to this extracellular domain, it is going to result into activation of smooth and receptor. Okay, so let me just point it down here. Let me just write it so that you will be able to understand it better. First is patch receptor. Okay, it is able to transport cholesterol molecule because it is actually a channel for a cholesterol molecule. Okay, so it is a large transmembrane protein which is residing over the primary cilium. Okay, so it is existing over the primary cilium part. It has ability to transport cholesterol molecule. Okay, and when it is active patched is going to transport cholesterol molecule, so which is going to result into decrease in the cholesterol concentration in plasma membrane. Okay, right? Whereas smooth and receptor, which is not present over the primary cilium, when it is when the patch is active, it is present over the cell membrane part, which is not a part of primary cilium. Okay, when the patch receptor is active, mind me. Mhm. So this smoothen receptor has extracellular domain which can bind to cholesterol, and the binding of this cholesterol to the extracellular domain of the smoothen receptor, that is going to activate smoothen receptor. Okay, which means this cholesterol binding activates the smoothen receptor. So this smoothen receptor, it is 7-pass transmembrane protein. Okay, which resembles to frizzled receptor. So let's see the scenario when the hedgehog is absent inside the cell. Okay, so this is the mammalian cell. This is the mammalian cell plasma membrane. This is the cell membrane of mammalian cell. So this hedgehog is not there. So hedgehog is absent. Let's assume that hedgehog is absent, and let's see what happens inside the cell. Okay, so well, I forgot to draw the primary cilium. So this is the So this is the plasma membrane and this is the primary cilium. Okay, and this is the plasma membrane well. So here you can see over the cell membrane part outside the primary cilium exists smoothen receptor, which is 7-pass transmembrane protein. Okay. So this 7-pass transmembrane protein has extracellular domain, which is able to bind to the cholesterol. Okay, so this particular extracellular domain has the affinity for the cholesterol molecules. So this is the smoothen receptor. Okay, while the patch receptor, which is present over the primary cilium, it is also a large transmembrane protein and it is present there. So when the hedgehog is absent, means the patch receptor will be active. So hedgehog is not there, so hedgehog cannot bind to this patch receptor. So this means that patch receptor is active. This means that this patch receptor is going to transport the cholesterol molecule. So it is going to transport the cholesterol molecule that were present here over the membrane, right? So that is going to decrease first. So before the patch was active, okay, let's say that the cholesterol was quite abundant. But now the patch is active because Hedgehog is not there. Due to the absence of Hedgehog, the activation of patch will be seen. Like this is the cell at rest. Okay, means the Hedgehog signaling is not there. So when the cell was at rest, when the Hedgehog was absent, this patch receptor protein was activated. Means it is going to remove those cholesterol molecules over from the plasma membrane. So the cholesterol molecule will be quite less. So they are very less. Like the abundance of cholesterol molecule is now less over the plasma membrane, and that is why smoothen will be inhibited. So patch receptor is going to inhibit smoothen. How? By controlling the availability of cholesterol on the plasma membrane. Because how does smoothen get activated? The extracellular domain, if it binds to the cholesterol which is present in the plasma membrane, so let's assume that these are the cholesterol molecule, and this binds to the extracellular domain of smoothen receptor, and then smoothen receptor will be activated. But now, if there is no cholesterol, then how the smoothen receptor will get activated? So this is how the patch receptors inhibit the smoothen receptor. Okay, so in the case of absence of Hedgehog, the patched receptor, it is going to inhibit smoothen receptor by controlling the abundance of the cholesterol on the plasma membrane. How? By controlling the abundance of the cholesterol on the plasma membrane when Hedgehog is absent. So when Hedgehog is absent, patched is active and smoothen will be inactivated. Okay, so this happens over the plasma membrane. Let's now see what happens inside the cell, inside the cytoplasm, in the cytoplasm, in the cytosol. So in the cytosol exist the gli transcription factors, the transcription regulators that are going to regulate the transcription of Hedgehog responsive genes. So when the Hedgehog is absent there, those transcription regulators will obviously be, they will be shut down. So so this means that the whatever genes, so let's assume that this is the nucleus and these are the genes, okay, which are the HH responsive gene, Hedgehog responsive gene. So if Hedgehog is not there, this means that Hedgehog responsive gene, they are repressed, they are in the repressed state. This means for the repression of Hedgehog responsive gene, for silencing of those Hedgehog responsive gene, we need certain repressor. So what are the repressor in this case? So the gli transcription factors that were present inside the cytoplasm, when the Hedgehog signaling is not there, they are being restricted. Okay, they are being restricted by a protein known as SUFU protein. Well, I did not list that down in the table above. So, well, I just explained that here. So, SUFU protein is a protein which is going to, which is a repressor. Okay, which is a repressor, or we can call it as a co-repressor, because actually repression job is done by gli proteins, because so gli proteins are the transcription regulators which will be transcription repressor in the absence of Hedgehog, and which will be transcription activator in the presence of Hedgehog. We'll see how. So how? Because of the presence of SUFU protein. So SUFU protein is suppressor of it is suppressor of fused. So this protein is co-repressor. Okay, so this co-repressor protein SUFU, let me draw it by a blue color. So this SUFU protein, it inhibits the gli transcription factor. So I already told you that this family has multiple gli proteins such as gli2 and gli3. So in this gli2, okay, it will be bound to the SUFU protein and it will be inactivated. Okay, so gli2 is bound to SUFU protein. So let's assume that this is gli2 and this is gli3. Okay, so this is gli2 and this is gli3. So both of them, they will be bound to SUFU when Hedgehog is not there. Okay, so this will inactivate gli2 protein, and this will be present in this form, SUFU bind form, inside the cytoplasm. So gli2 will bind to SUFU and it gets inactivated. so that it cannot go and act as a transcription activator, and that is why HH responsive gene will be silent. Whereas GLI3 protein, it also binds to SUFU protein, and what happens here is that GLI3 protein is going to act as the repressor. How? When it binds to the SUFU protein, the this certain certain kinases can work on this GLI3 protein, such as protein kinase A specifically. Okay, so protein kinase A works on this SUFU bound GLI protein to break this GLI protein down into a smaller portion, which acts as a repressor, and some other part. So here you can clearly see I have drawn a smaller part and a bigger part. So due to the phosphorylation by pKa, this superbound GLI protein will undergo proteasomal degradation, right? Because due to the phosphorylation of GLI3, it will be subjected to then ubiquitination, and that is going to result into proteasomal degradation of GLI3, and this GLI3 will be broken down in a smaller part and in a larger part. So larger part will be totally degraded, and this smaller part, it will work as a repressor, which is referred as GLI3R. So this repressor, okay, so this repressor, GLI3 repressor, oh, not GLI2, GLI3 repressor. So this small part and the larger part, it is actually cleaved completely, it is degraded completely. So the smaller part will act as a repressor. So this is GLI3 repressor, and it is going to translocate inside the nucleus, and it will bind to the HH responsive gene, and it is going to inhibit the transcription of HH responsive gene. So this happens when the hedgehog is not present inside the in the biological system. So how does, so I told you that GLI3, it binds with SUFU. So SUFU protein is a kind of scaffold protein which is going to provide a platform for attachment of the kinases such as pKa or CK1 or the GSK3, which will be phosphorylating GLI3 protein. So let me just zoom in this scenario a bit. So what will happen? So this is the GLI3 protein. Okay, this is GLI3 protein. GLI3 protein, which is bound with SUFU protein. So let's assume that this is SUFU protein. Okay, so the SUFU protein, it provides a platform for the attachment of kinases. So now the kinases such as pKa can adhere to this complex. The kinases such as CK1, the kinase such as GSK3, they can adhere to this complex, and they can now phosphorylate this GLI3, and phosphorylation of GLI3 will result into ubiquitination, which will result into proteasomal degradation. But did did you have this question that how does this pKa is going to get activated? So this gets activated due to other protein, okay, which is a G protein known as GPR161, which is present on the plasma membrane. So this is actually a stimulatory G protein. So this is going to stimulate adenylene cyclase, which is going to increase the CAMP levels inside the cell. I hope you have followed this. I'm assuming that you have been following my cell signaling series from the start. So if you have been following, you won't be confused in this particular part. So when the CAMP increases inside the cell, the pKa, protein kinase A, gets activated, right? So how is protein? So this could be a potential question, that how this pKa gets activated in the hedgehog signaling. So pKa gets activated due to the activation of a G protein known as GPR161. So this is how all of the different signaling pathways, they are quite interlinked. So this G protein, GPR161, it is actually a stimulatory G protein, which is going to stimulate the the adenylyl cyclase, which will convert the ATP to CAMP, cyclic AMP. So increase in cyclic AMP level will result into activation of protein kinase A, which will be recruited over this particular complex. So this complex is known as the repressing complex, because this SUFU protein is going to, so that is why SUFU protein is going to act as a co-repressor, because due to this SUFU protein, the GLI3 protein is getting converted into GLI3 repressor protein, which is going to transmigrate into the nucleus, and it is repressing the the hedgehog responsive genes. Okay, so I hope the scenario when the hedgehog is absent is clear to you all. So let's now discuss what happens when hedgehog is present in the system. So when hedgehog is present in the mammalian cell, what happens? So this is the plasma membrane part, okay, and this is the primary cilium. Okay, so when hedgehog is present, the patch receptor will bind to hedgehog and it will get inactivated. So this patch receptor will migrate out of the primary cilium to the cell membrane part, which is not a part of primary cilium. So this patch receptor, it will bind to hedgehog receptor. Okay, so this is hedgehog receptor, and it is bound to the patch receptor, which will inactivate the patch receptor. So this is the inactivated patch receptor. Okay, so binding of HH to the patch receptor will result into inactivation of patch receptor. So patch receptor, which was previously active and it was transporting cholesterol molecules out of the cell membrane, will now be inactivated, okay, due to the binding of hedgehog, and that is why now the cholesterol, it will be present abundantly in the plasma membrane, because it is not being kicked out by the this patch receptor, which was actually a transporter for the cholesterol. So this cholesterol is abundant in the plasma membrane, and that is why now this cholesterol is going to bind to the smoothen receptor. Okay, the smoothen receptor, let me just redraw it. Okay, so the smoothen receptor, which has extracellular domain, which can be bind to the cholesterol. So this smoothen receptor will now bind, it will bind to the cholesterol present on the plasma membrane, and that is going to activate the smoothen receptor. Okay, so now the smoothen receptor will be activated in the presence of the hedgehog, because the patch receptor is being inhibited. So this activated smoothen receptor, it is going to result into the activation of the GLI transcription factor. How? Because the smoothen receptor will now recruit the SUFU proteins. So this SUFU protein will get attached uh, to this activated smoothened protein. So this SUFU protein binds to the, or it gets occupied with the smoothened protein. So this, so now as SUFU protein is preoccupied with smoothened protein, so this engagement of SUFU and smoothen will result into release of GLI transcription factors. So this will result into release of GLI2 and GLI3. So let me just change the color. So this will result into release of GLI2 and GLI3. Okay, so this GLI2 and GLI3 will get activated. So GLI3 does not do this job. So actually the SUFU protein, which was bind to the GLI2, only that SUFU protein will be engaged with smoothen protein, but the SUFU protein will still be, it will be still adhere to the GLI3 protein. Why does this happen? Well, this is because this smoothen activation, it is done due to the cholesterol. Okay, so this is going to recruit the SUFU proteins, but this is also due to the phosphorylation of smoothen. So that multiple phenomena is going to activate smoothen protein to recruit SUFU protein, which was initially bind to GLI2 protein. But the GLI3 protein bound SUFU protein will still stay in the bound form, so that this GLI3 protein will not get it will not get, it will not get phosphorylated and by pKa and CK1 and GSK3, because they are already preoccupied with the smoothen protein, with the phosphorylation of smoothen protein over here. So SUFU protein will release GLI2, and this GLI2 will now go into the nucleus to activate the HH responsive gene. So HH responsive gene will activate and they will get transcribed. Okay, so this is how the activation of smoothing protein will result into the activation of GLI proteins that is going to result into the transcription of Hedgehog. So this is the scenario in the case of mammalian cell. But in the case of Drosophila, the proteins that are going to engage in this signaling pathway, they are different. For example, the cubitus interruptus is the transcription regulator in case of Drosophila. So in the case of Drosophila, the primary cilium is not formed. Cilium is absent. Okay. So instead, on the plasma membrane exists the patch receptor. The smoothen receptor is present over the vesicles inside the cytoplasm. Okay. And there exists IHOG receptor. So this is IHOG receptor, which is a co-receptor for Hedgehog. So when Hedgehog comes, it is going to bind to both this patch receptor as well as IHOG, and it is going to form a co-receptor dimer, which is going to result into activation of smoothened. So when the hedgehog is not there, okay, when hedgehog is not there, the smoothened will be inhibited, and that is going to result into the cubitus interruptus, which was present inside the cell. So what will happen? The hedgehog, when is not there inside the environment in the biological system, the cubitus interruptus, which was present inside the cell cytoplasm, it is going to get phosphorylated by pKa, GSK3, and the CK1, casein kinase 1. So all of them, they are going to phosphorylate cubitus interruptus, which is going to result into the tagging, tagging this cubitus interruptus by ubiquitin molecule. So this is going to result into the ubiquitination. So first happens the phosphorylation event and then the ubiquitination event, which will expose this ubiquitinated cubitus interruptus to the proteasomal degradation. So proteasomal degradation will be done, and that is going to result into the cleavage of cubitus interruptus. That is going to be cleaved into two parts. The smaller part is known as cubitus interruptus smaller protein, which is going to work as a repressor. So when this cubitus interruptus it is degraded, so it is going to act as a repressor, and that is going to repress the whole HH responsive gene in the Drosophila. So in the Drosophila, primary cilium is absent, but instead of primary cilium, the smoothened receptors, they are being internalized by keeping them on the vesicles. Okay, so this is the first difference in the Drosophila. The other difference is the presence of cubitus interruptus instead of GLI proteins. So the Drosophila has cubitus interruptus, and the kinases that are going to work in the Drosophila are pKa, GSK3, and CK1. In the case of mammalian cells, only pKa works, but sometimes, or if the cell type is different or the developmental stage is different, GSK3 and CK1 can also work. So the other difference between the mammals and Drosophila is that instead of SUFU proteins, the protein that is going to act as the adapter for the phosphorylation of the transcription regulator, the proteins that are going to work as adapter proteins are fused and COS2 or coastal 2. So fused and coastal 2, they work as co-repressor instead of SUFU in the case of mammalian cell. Okay, so there exists one more difference, that instead of SUFU, the Drosophila cell has fused and COS2 proteins. Okay, so these are the differences between the Drosophila hedgehog signaling and the hedgehog signaling that is seen in the mammalian cells. In the Bruchselber, there is a schematic about the whole signaling pathway that is done in the mammalian cell. So this is how the hedgehog signaling goes on in the mammalian cell. So when the hedgehog is not there, when the hedgehog is absent in the biological system, the smoothen receptor, it is being inhibited by the patch receptor. So here you can clearly see this is the primary cilium, and over the cell membrane of the primary cilium, the patch receptor is present. So this patch receptor, it inhibits smoothen receptor by transporting all of the cholesterol molecules that are present on this membrane part. So if the cholesterol is absent, that cholesterol, the plasma membrane cholesterol, cannot interact with the smoothen receptor. Well, because it is absent, it is being transported either in the extracellular matrix or in the endosomes by the activated patch receptor, and that is going to inactivate or inhibit the smoothen receptor. So this is going to result into the activation of the G protein GPR161, which is present again over the cell membrane part of primary cilium. That is going to result into the activation of adenylyl cyclase to increase cyclic AMP levels inside the cell, which will activate a protein kinase A, PKA. So this PKA works over the GLI3, which is bind to SUFU. So as said, the GLI2 protein and GLI3 protein, they belong to GLI family of transcription regulators, and both of them, they are bound with SUFU protein, which is also a co-repressor when the hedgehog is not there. So when hedgehog is absent, the SUFU will bind to those GLI transcription receptors. So GLI2 will be inactivated just by the binding to the SUFU, while GLI3, which is bound to the SUFU, in this case, the SUFU protein works as an adapter protein, and it helps the recruitment of the kinases such as protein kinase A over this GLI3. So it is going to form a whole complex which is going to promote the phosphorylation of GLI3. So this phosphorylation of GLI3, here you can see the GLI3 is being phosphorylated by PKA. This is going to result into the proteolysis of the GLI3 protein, which is going to result into the formation of GLI3 repressor and the dissociation of SUFU from GLI3. So here you can see SUFU is dissociated from the GLI3 protein, and the other part of GLI3 molecule, it is being degraded. So this GLI3 previously I said the smaller part, well, the smaller part is being degraded, while the larger part, it works as a repressor, and this transmigrate or translocates inside the nucleus, and it is going to switch off the hedgehog gene by acting as the repressor molecule. So this is how, in the absence of hedgehog, what happens? Patch receptor is activated, which is going to inhibit smoothen, which is going to result into the proteolytic cleavage of the GLI3 receptor, which will result into formation of GLI3 repressor, which will switch off or silence the hedgehog responsive gene. But in the case when the hedgehog signal is present, the hedgehog molecule, they are going to get bind to the patch receptor, and that is why this patch receptor will migrate in the cell membrane part outside the primary cilium. So the binding of hedgehog to patch receptor is going to inactivate the patch receptor. So that is why the transport of the cholesterol molecule will be stopped. So now the transport of cholesterol molecule is not being done. That is why cholesterol is quite abundant in the plasma membrane, and that is why this particular cholesterol will bind to the smoothen receptor, and that is going to activate the smoothen receptor. So this activated smoothen receptor will result into dissociation of SUFU, which was previously bind to GLI2 protein. So this is going to result into release of GLI2 protein in the cytoplasm, and well, GLI3 is still bound to the SUFU protein, but as PKA and CK1 and GSK3, they are preoccupied with smoothened, they are not going to work on this particular complex, the GLI3 and SUFU bound complex, and that is why this complex will stay as it is in the cytoplasm, and this is not going to act as a repressor. Whereas the SUFU, which was bound to the GLI2 protein, it is going to release the GLI2 protein inside the cytoplasm, and now this activated GLI2 protein, see here you can see that it is going to be migrated in the cytoplasm with the help of the microtubules. So here you can see the importance of microtubules, how it is transporting the signal molecules from the tip of primary cilium towards the intracellular part. So it will transport the GLI2 protein towards the intracellular side to the cytoplasm, and this activated SUFU protein will migrate to the nucleus to work as a activator. So it is going to work as an activator and it is going to activate the Hedgehog signaling. So this is how the Hedgehog signaling pathway is mediated in the mammalian cell. Well, there's one more question. In the case of Drosophila, here you can see in the case of mammalian cell, the patch receptor and the smoothen receptor, both of them they are present on the cell membrane, which is which is a part of the plasma. So they are present on the plasma membrane, which is connected. So they are present on the single membrane, right? But in the case of the Drosophila, the membranes where the patch and smoothen receptor that are present, those membranes are different. So in the case of Drosophila, patch receptor is present on the cell membrane, whereas the smoothen receptor is present on the vesicular membrane of the vesicular membrane, right? So the lipid bilayers where the smoothen and patch they exist, they are very different. So the exact mechanism by which the patch receptor is going to inhibit smoothen receptor, it is not quite clear. I mean, I need to read literature about that. I'm not sure how patch receptor is going to inhibit the smoothen receptor in the case of Drosophila. In the case of mammalian cell, the mechanism is somewhat clear, because the cholesterol, it is regulating, the patch receptor is regulating the abundance of cholesterol in the membrane. But how it is able to manage the abundance of cholesterol in the case where the cell membranes are quite different? So we can say that, okay, patch receptor is going to decrease the cholesterol abundance of the cell membrane, but smoothen is not present there, right? Smoothen is present on the vesicular membrane. So how this patch receptor is inhibiting smoothen, that is not quite clear to me yet. So I need to read the literature. If you have any idea about this, you can pin that in the comment section below. Or if I by somehow I get it, I get an idea how the patch receptor is going to inhibit the smoothen receptor. If I have the idea about the mechanism, I'll post it in the description box below. So with this I conclude the lecture. In next lecture, we will be discussing about NF-kappa based signaling, which is again a different type of signaling, and that will be the last lecture, and we'll be done with the cellular signaling part. So till then, see you. Bye bye.